Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem about this lesson. So here we will discuss about the photophosphorylation and Calvin cycle. So that is all thing is related to the photosynthesis. Photosynthesis mean the light, using the light to produce sugar. So that's whole mechanism is depend to the photosynthesis in the presence of chlorophyll and carbon dioxide and water, which that produce the sugar. So here is the photosynthesis is light dependent and light independent reaction composition to produce sugar. So first of all, light dependent reaction produce ATP and NADPH, while light independent reaction produce sugar in the form of 3GP. So that's G3P. Glyceraldehyde 3 phosphate will be produced 3 carbon containing molecule in the Calvin cycle dark reaction. So let's talk about the major concept about the cell where the Calvin cycle and light reaction is occur. So here is the cross section of the leaf which they are showing. Inside the vascular bundle contains xylem and phloem and surrounded by the bundle sheath cell. And there are many spongy mesophyll cell as well as palisade mesophyll cell are present inside. So these spongy mesophyll cell is contain air spaces, while palisade mesophyll cells is not contain spaces. So below that is a stomata. So the stomata is the made up of guard cell. So the guard cell is outer, and that is produce a pore-like structure is known as the stomata, which that is open and closed on the basis of condition. So here is the carbon dioxide and oxygen exchange. For example, oxygen will release while the carbon dioxide will be inhaled inside, move inside into the mesophyll cell for synthesis of glucose, and that is the photophosphorylation and Calvin cycle to produce. Let's talk. So here is I am drawing the one cell which contain nucleus, cytoplasm, chloroplast as well as mitochondria, and there are many things. On the upper side is the cell wall is present, while inside is the cell membrane is also present. So vacuole chloroplast. So the chloroplast is the double membrane organelle. So the double membrane organelle which that is used for the light harvesting system, which that use the light to produce NADH and ATP, as well as to produce sugar. So here is the light which that is coming toward the chloroplast is the organelle which contain a thylakoid membrane inside. So here I am drawing the chloroplast as an organelle which contain a double membrane organelle which contain a thylakoid. So these thylakoid contain a different stack-like structure is present, which that is looking like this. For example, coin to coin, and that is make a column. So this is the granum. Is single is granum and plural is the grana, and that is the stroma is present inside is the which contain a Rubisco enzyme, which that is the Rubisco is the worldwide is highest protein quantity in the worldwide because all plant. So here I am giving the cross section of the granum which contain a different complexes as well as photosystem. So these photosystem contain chlorophyll. So this chlorophyll is absorb the light, that is absorb the light and move the electron. That's electron basically light absorb one by one, and this here we will discuss whole detail. So first of all we should understand the concept. Here is the molecule of water which are split into the 2 proton 2 electron as well as half molecule of oxygen. So lumen is lumen is present inside, while the others outside is the stroma. So that is the cytochrome complexes are present, which that is flow the electron and electron, which means that electron is helping by the cytochrome complex to move forward, and this forward moving is irreversible because it's losing energy for moving backward. So that's further going on. So the photosystem 1 and photosystem 2 contain first of all photosystem 2, which that is use light 680 nanometer, and photosystem 1 which contain 700 nanometer light, which that is absorbed in the wavelength. So that is the NADPH reductase enzyme and ATP synthetase enzyme are present as a protein-like structure. So let's talk about all things is here. So the first of all, I want to say that the plant is the autotrophs, that living organism who makes their food by themselves by light harvesting, and photosystem contain space chlorophyll pigments. That's basically the chlorophyll pigment containing a plant is the autotrophs. So this autotroph means produce energy itself by the using of light. On the other hand, heterotrophs, which that is the animal which that consume the energy, consume the food from the autotrophs. So the auto means self, hetero means the different to another. So here is the light which that is used for it. Next, we will discuss about the whole mechanism in the thylakoid membrane, and that is where the lumen and the cell membrane is basically here. So this is the thylakoid membrane, is looking like this, and this thylakoid membrane contain many complexes. For example, first of all, we will discuss just remember about that the Z scheme model, which that is look like this. But we will understand about the easy to understand the Z scheme model, which that is same like a photosystem 1 and photosystem 2. So here is the stroma, and here is the thylakoid space, which contain a high concentration of proton. So that remember about that. So here is the photosystem 2. Photosystem 2 contain a photon, which that is the range of wavelength is 680 nanometer. So here is the green color, which that is showing in the figure. Green color is the chlorophyll containing protein. So this chlorophyll containing protein is moved from basically from primary to the basically one by one molecule to another to lose their energy and ultimately accept the 680 nanometer containing wavelength in the uh, in the in the antenna complex. So here is the antenna complex which that is used. So this is all is contain a chlorophyll that is known as the primary acceptor of electron. So this is the photosystem 2, and that is the electron acceptor. So accept the electron by the hydrolysis. That's water split into the oxygen and proton, and 2 electron will be received by it. So the primary acceptor will be, and that's excite of the electron due to the excitement of the electron. Black line shows that the transfer of energy, and the black dot is the black circle, it shows that the acceptor of electron. Then when accept the electron from the water, and that means this is a primary acceptor, and this primary acceptor of electron will be further go into the plastoquinine. So this the plastoquinine is the another protein part are present in the thylakoid membrane, and these accept the uh, four four proton, and 4 proton is accept from the plastoquinine, and basically that's a 2 electron will be receive. So the 2 electron will receive from the plastoquinine, and that is the 4 proton will be move inside. So the 2 electron donate to the cytochrome complex BF. So this cytochrome complex BF is accept the 2 electron, while the 4 proton will be moved inside of the thylakoid membrane. So remember about that is stroma will be decrease the proton level, while increase the level of proton inside of the thylakoid membrane, inside of the space inside. So is the plastocyanine will be accept these 2 electron, and there's 2 electron of the plastocyanine will be transferred to the photosystem 1, which that's contain a 700 nanometer light wavelength, which that is the light transfer of energy again from the light, and this light will be transferred one by one and accept that again electron. So what is the meaning of this? Electron was losing the energy from a photosystem complex to photosystem 2, and when losing this energy, that is ultimately again will be excite. So that's the primary electron acceptor will be after the excitement of the electron. After the excitement, it means the again reserve the energy as compared to the middle where the plastocyanide, which contain a less amount of energy for accepting the electron. So the photosystem 1 contain a 700 nanometer light, which that means it means the again more energy will be produced from the light, and that's electron will be again excite, and that's primary electron acceptor will be accept this prime electron, and that's go into the secondary mechanism ferrodoxin. So this is the ferrodoxin is the protein which iron containing protein which that's accept the two electron. For example, that's accept the two electron, and these two electron is basically go into the NAD plus reductase. NADP plus reductase or NADH reductase, which that the. So here is the photosystem 1 which that donate the electron to the FD ferrodoxin, and this will be reduced into the ferrodoxin, and that is basically ferrodoxin ion will be formed, and that's negative charge, and it means the NAD plus reductase enzyme is here, which that is the thylakoid membrane containing NAD plus reductase enzyme, which that is reduce the NAD plus into NADH. It means the stroma will be decreasing the proton concentration, while inside is the more proton. It means the higher concentration inside as compared to the outside. How it's possible? So here is the NADPH will be formed. It means the decreasing of the proton, and 4 hydrogen ion will be moved into the lumen, while 2 proton is already produced from the water. So hydrolysis will produce 2 proton, and 4 proton will become from the outside. So that is mean that it's going is the use as a chemiosmosis. So here I am drawing that is the chemiosmosis is basically here, that is the ATP synthetase enzyme is the ATP synthetase pump are present is here, which that's contain a different region F0 and F1. F1 contain a static, and F0 contain a rotator. So there are many part is the sub monomer, and there are the many monomer containing a complex units. Here is the epsilon, gamma, and B unit, alpha, beta unit, and that is the basically gamma and epsilon. That is the here. So what will be occur? Here is the channel like structure. Look like that. The blue color shows that the proton. This proton is filled inside from the F0 region, and this filling is moved into the stroma, and it's how it's possible to move. That mechanism is we will discuss. Electron will move from here, that is the channel like structure, and this electron is fixed into here. It means that is the monomer of the electron proton acceptor, and that here is the proton will be enter inside. So it means the empty space of the proton acceptor site is accept the proton, and the one proton will be released due to the concentration of high proton. So that's a 2 proton will not live in this region. So it means move into the epsilon, gamma to beta unit monomer, and that means proton will be excrete move outside. So it's the static, and here is the rotator. It means a rotation. Rotation causes the movement of proton down into the stroma. So the 4 proton is required to make our synthesis ATP from the ADP. So the joining of the phosphate group is due to the 4 proton. So important to know that here is the photophosphorylation, the using of light to synthesis of ATP from ADP, and it means that basically ATP that is known as the photophosphorylation. Why? Because it is using the light. So that means this ATP is produced due to the using of light. So this reaction is known as the light reaction and light dependent reaction. While the other dark reaction we will discuss in the further in this video. So here that first of all we will discuss about the calculation of the proton. So here the water which that split into the 2 proton, 2 electron and half oxygen. So the 2 molecule of water will be released the 1 molecule of oxygen. So here is the 12 basically 2 electron is used, that is a proton pump into the lumen, is the 4 proton is pump inside from the plastoquinine binding, and that is move inside, while total proton is the basically 6 proton inside due to the 2 is come from the water and the 4 is come from the proton pump, which that is a move inside the lumen due to the electron flow. Okay, so here is the NADH, which there is a 1 proton is the come from the outside, so it means we are not including in it, while we can include the 2 electron. So the outside proton and 2 electron is used to produce NADH, and that is acidic medium will be caused due to the proton. Okay, next I hope you make sense about these things. We are understanding the proton calculation. So here is I want to describe the light reaction ATP NADPH. This is the production of light reaction, while these production ATP and NADPH is go into the stroma, and that is there are many more number of ribosco compound or ribulose bisphosphate are present. This RUBP is the protein content, and that is used as a enzymatically. And here's the dark reaction of light independent reaction. A dark reaction means the light is not depending, but it's depend by the passively, not actively, because passively mean the light reaction give the ATP and NADPH. So let's talk about here is the basically carbon dioxide and oxygen is exchanged. So the carbon dioxide move inside into the plant, so go into the cell. So here is the ribulose bisphosphate or RUBP, which that is accept of the 3 molecule of carbon dioxide. It means the 3 carbon containing compound. So these 3 molecule of RUBP which receive the 3 carbon containing molecule, so is convert into the 6 molecule of 3 phosphoglycerate. So there's conversion. It means the 6 molecule. So here is the 6 ATP used and 6 ADP convert. So it means that 6 ATP is used in this time, while next we will discuss is the 3 ATP is more used, where we will discuss. So that is 1,3 bisphosphoglycerate is formed. 1,3 bisphospho means the 6 molecule of this. It means the carbon number 1 and carbon number 3 contain a phosphate group, while here is the NADH. NADPH is used to produce NAD plus, so that is the reductase reduction, and phosphate group is oxidized. So that's a phosphate group release 6 phosphate group release and form a glyceraldehyde 3 phosphate G3P. So this G3P will be separate 1 molecule of G3P, while the 5 molecule of 3 carbon containing G3Ps move for regeneration. So that is important to know that. First of all, we had discussed about the carbon fixation, and the next is the reduction, and here it was the regeneration for it. So Remember about that. Here is the phase 1, which use at the carbon fixation, and here is the phase 2, which that is used for as a reduction, and that is the here is the regeneration phase, which that is the RUBB, again regenerate. So that is the again regeneration to make a 5 molecule to convert 3 molecule of RUBP, and this cycle is again. So it means the 1 molecule of G3P will be formed and will be separate, while the 5 will be again move. What that mean? It means one molecule will be separate. It means the 2 turn of the Kelvin cycle produce the 2 molecule of G3P to produce glucose. So right. So here is the 3 carbon dioxide can be moved more add for double turning to produce sugar. So sugar is the 5 carbon containing molecule, which that use as a energy in our body as well as the plant also. So here is the sugar. So first of all, we will discuss about the basically here is the 3 molecule of ATP is used here, and here is the basically 3 molecule, and 9 total will be used, and 6 NADPH is used to produce 1 G3P. So it means double will be 12 NADPH and 18 ATP will be formed. How it's possible? Next, here we will discuss about the calculation of proton to understand it. Right? So here I am drawing that is the 12 hydrogen, 12 water molecule is produced 12 NADH. Why? It means that 2 electron produced. So it means the 2 electron mean the 2 electron produce 1 NADH. It means the total is the 12 H2O produce the 12 NADH, because the 2 electron is produced from 1 molecule of water. So here the 12 water molecule will be multiplied by the 6 proton to produce 72 proton. It means the 1 molecule of water will produce the 6 molecule of proton. So multiply, and here is the basically 6 is into the lumen for produce ATP. So that's why is here that is the 4 ATP per ATP, 4 proton per ATP to produce 18 ATP due to the divided by 4. So that's here is the basically 1 water molecule produce the 1.5 ATP due to the 4 proton production. So 4 proton will be released into the stroma due to the lumen to stroma to produce 1.5 ATP, because 6 proton will be formed. So next here is the photosynthesis. So this photosynthesis equation is quite easy to understand now. That is the 12 H2 and 6 carbon dioxide, and that is the basically to produce sugar. So thanks for watching.